Hello! I'm going to be reading a paper published in the Annual Review of Immunology titled The Interplay Between Innate Immunity and Non-Immune Cells in Lung Damage, Inflammation, and Repair. This was published in 2025, and I will leave a link if anyone would like to read along with me, but if not, it is okay. I will just be reading this. So, let's get started. So, starting off with keywords: epithelium, endothelium, macrophage, infection, regeneration, fibrosis. Abstract. As the site of gas exchange, the lung is critical for organismal survival. It is also subject to continual environmental insults inflicted by pathogens, particles, and toxins. Sometimes these insults result in structural damage and the initiation of an innate immune response. Operating in parallel, the immune response aims to eliminate the threat, while the repair process ensures continual physiological function of the lung. The inflammatory response and repair processes are thus inextricably linked in time and space, but are often studied in isolation. Here, we review the interplay of innate immune cells and non-immune cells during lung insult and repair. We highlight how cellular crosstalk can fine-tune the circuitry of the immune response, how innate immune cells can facilitate or antagonize proper organ repair, and the prolonged changes to lung immunity and physiology that can result from acute immune responses and repair processes. Introduction. Among the barrier of organs in mammalian organisms, the lung may have to do the most delicate balancing act of all. On one hand, a vast and highly permeable surface is required for efficient gas exchange to supply the organism with oxygen and to discard carbon dioxide. Estimates for the human adult lung indicate values of approximately 100 meters squared of exposed surface. On the other hand, inhaled air carries particles, toxins, pathogens, and other substances, and constant surveillance is required to monitor for potential infection or harm. Sophisticated sentinel systems are at work, and host responses to harmful agents tightly controlled to limit damage as far as possible. As most mammals can survive without oxygen for only a few minutes, lung functionality must be guaranteed at all times during these host responses. Furthermore, the capacity for gas exchange must be fully restored after lung insult to avoid progressive functional decay over a lifetime. In this review, we highlight how interactions between innate immune response and epithelial, stromal, and other non-immune structural components crucially contribute to keeping the balance in the tightrope walk between lung damage, protection, and repair. The steady-state lung and an anti-inflammatory environment. To limit inflammation-driven interference with gas exchange, the healthy lung is equipped with mechanisms acting at several levels to keep inflammatory responses to a minimum. In this section, we introduce the main cellular constituents of the lung and how they contribute to its steady-state quiescence, I think is pronounced. More comprehensive accounts of all cell types present in the lung are being developed by several consortia of lung cell experts, and single-cell data are available at several lung atlases. See related resources. The actual interface between the organism and the environment is formed by epithelial lining the lung cavities. They act as physical and immunological barriers that constantly sense, react to, and adapt to external signals, protecting the organism against pathogens as well as mechanical and chemical damage. Two different epithelial types are found in the lung: the airway epithelium, which is pseudostratified columnar epithelium consisting mainly of basal, ciliated, and secretory cells, and the alveolar epithelium, which is distinct in structure and development from the proximal airways and constitutes the functional units of the lung where gas exchange occurs. Alveolar type 1, AT1, cells cover the vast majority of the alveolar surfaces, closely juxtaposed to the capillary network, and are responsible for oxygen uptake. Cuboid alveolar type 2, AT2 cells produce and recycle the surfactant proteins and lipids required to regulate surface tension inside the alveolus. Beyond having a physiological function to permit ventilation and gas exchange, the epithelium also provides a first line of defense against insult. Tight junctions between cells contribute to the establishment of a physical barrier to pathogen and particle entry, while the production of mucus by goblet and other secretory cells, combined with the ciliary beat, is essential for the mucociliary clearance of pathogens and debris. The importance of this process for lung homeostasis is evidenced by chronic lung disease that results from both mucin hyperproduction and ciliary dysfunction. The mucus layer not only is a vector for the physical evacuation of pathogens, but also contains antimicrobial components such as secretory IgA, which is immunoglobulin, I'm so bad at saying that word, antibody. It's a type of antibody, immunoglobulin A, cytokines, and lysozyme. The production of surfactants by AT2 cells in the alveoli also plays an important role in immune homeostasis. For example, phospholipids present in surfactants can antagonize ligand-induced activation of toll-like receptors, TLRs, such as TLR2 and 4, and interface with the binding of viruses to host cell plasma membrane receptors. Relative to other barrier tissues, such as the gastrointestinal tract and the skin, the airway and alveolar regions of the lung exhibit slow cellular turnover and proliferation during homeostasis. This quiescence is regulated by complex crosstalk between epithelial and underlying mesenchyme. Specifically, the airway epithelium actively maintains mesenchymal quiescence through paracrine sonic hedgehog signaling, which in turn regulates a feedback loop to maintain epithelial quiescence. Perturbation of epithelial mesenchymal crosstalk could disrupt the normal quiescent state of both cellular compartments in the absence of damage, highlighting the importance of cell-to-cell contact to regulate the proliferative response. Furthermore, interactions between epithelial and mesenchymal cells involving WNT signaling contribute to the generation of localized private stem cell niches that are required to maintain and restrain both basal cells and AT2 stem cell function at homeostasis. Capillary endothelia lie closely juxtaposed to alveolar epithelia to facilitate gas exchange. In non-inflamed tissue, endothelial barrier integrity is tightly regulated, allowing for a controlled exchange of fluids, macromolecules, and cells between the circulation and tissue. Under normal conditions, this regulation is due to endothelial control of several cellular and physiological aspects of circulation. Endothelia control blood fluidity via the production of coagulation inhibitors, blood clot lysing enzymes, and a glycocalyx with an anticoagulation properties, as well as blood flow via production of vasoreactants such as nitric oxide and PGI2 that act on underlying smooth muscle cells. The endothelium also regulates vessel permeability in a dynamic manner via the presence of intracellular, tight, and adherent junctions that seal off the pericellular space between endothelial cells, ECs. Finally, the endothelium orchestrates the quiescence of circulating leukocytes, as resting ECs show minimal expression of adhesion molecules such as P/E selectins, VCAM, and ICAM1 on their surface. In addition to this regulation at the transcriptional level, the presence of a well-developed endothelial glycocalyx may also provide steric hindrance to leukocyte contact with endothelial adhesion molecules. In association with lung epithelia lie resident alveolar macrophages, AMs. The resident Res-AM, population is established during embryonic development, in concert with lung organogenesis and alveolarization. AMs are indispensable for the regulation of surfactant levels. They catabolize and recycle surfactants produced in large amounts by alveolar epithelial cells. This essential function is facilitated by a bias towards lipid metabolism, reinforced by the transcription factor peroxisome proliferation activator receptor gamma, PPAR gamma, which is upregulated in response to granulocyte macrophage colony stimulating factor, GM-CSF, from AT2 cells, and autocrine transforming growth factor beta, TGF-beta, TGF-beta receptor signaling, and is essential for AM survival and function. Accordingly, absence of AMs, loss or neutralization of GM-CSF, and loss of PPAR gamma result in pulmonary alveolar proteinosis, a disease of pathological surfactant accumulation. Notably, AMs are the only immune cells whose absence at steady state results in lung disease. Another important role of AMs is to maintain alveolar order by recognizing and clearing apoptotic cell debris and extracellular matrix (ECM) deposits via efferocytosis, which is evidenced by the rapid accumulation of dead cell components in their absence at steady state. Efferocytosis contributes to limiting the inflammatory response of AMs due to their expression of TAM receptor family tyrosine kinases such as AXL and MERK-TK, which activate suppressor of cytokine signaling 1, SOCS1, and SOCS3, in turn inhibiting TLR and cytokine signaling pathways. Expression of the transcription factor KLF4, Krepel-like factor 4, by AMs downregulates expression of TLR9 and thus allows for clearance of apoptotic cells while limiting inflammation caused by DNA sensing. However, the homeostatic function of AMs is to maintain not only the physiological integrity of the lung, but also its immunological steady state. AMs must effectively balance the requirement of efficiently clearing insults while maintaining a high threshold for initiation of an inflammatory response to prevent unnecessary inflammation or damage in response to qualitatively or quantitatively innocuous insults. With only one AM per 3 alveoli at any given time, AMs have a substantial migratory capacity that allows them to move between alveoli through the pores of con. This activity allows AMs to conceal pathogens from recognition by other cell types, which may contribute to excess inflammation. Furthermore, it may shield other cell types from productive infection that will result in dissemination of the pathogen. Indeed, impairment of AM migratory capacity or administration of clonorodate lysosomes to deplete the AM pool results in excessive neutrophil recruitment upon bacterial infection, while, in the context of low-dose viral infections, AM death or depletion results in higher viral load within the surrounding viral epithelium. The lung also harbors interstitial macrophages, IMs, a population of macrophages that reside in the lung interstitium. Recent studies have identified subsets within the IM compartment that exhibit distinct surface marker expression, chemokine profiles, and spatial localization. Numerous studies have identified a subset of nerve-associated IMs defined as CD206 negative, MHC2 positive, as well as CD169 positive. Another subset defined by CD206 positive, MHC2 negative, was also found to express high levels of LIVE1 and is found primarily surrounding the vasculature. The exact roles that these subsets play at steady state remain to be elucidated, but their occupancy of relatively well-defined niches, which can be found across multiple organs, suggests that they may fulfill important roles therein. While canonically associated with the inflammatory response, the steady-state lung harbors a population of neutrophils known as the marginated pool. These neutrophils are located to the lung capillary bed and interact with the circulating pool of neutrophils. This population can be rapidly released into the bloodstream or recruited into the interstitium and is actively maintained by the constitutive expression of CXCL12 by the pulmonary endothelium, which interacts with CXCR4 on neutrophils. The marginated pool is also thought to synergize with the capillary endothelium for general bloodstream pathogen surveillance. In a model of Escherichia coli bacteremia bacilli were rapidly immobilized along the vascular endothelium, followed by TLR4 and CD11b dependent neutrophil chemotaxis, resulting in pathogen uptake and elimination. Other immune cell types that are resident in the steady-state lung are innate lymphoid cells, ILCs, mainly of the type 3 subset, ILC3s, in humans, while ILC2s predominate in mice. Like some macrophage subsets mentioned above, ILC2s can associate closely with neurons in the lung. The lack of steady-state lung phenotype in ILC2 depleted mice suggests that ILC2s have no major homeostatic role, but that rather they are part of the lung sentinel system and able to respond rapidly to lung insults. T and B cells of several subtypes along the naive effector memory regulatory tissue resident spectrum are also present in the steady-state lung. This is more pronounced in humans than in mice, perhaps due to the much cleaner lifestyle with lower exposure history of laboratory mice compared to humans. The lung also harbors conventional monocyte-derived and plasmacytoid resident dendritic cells, which contribute to airway surveillance by continuously sampling the lumen for antigens, as well as a population of basophils, whose functions include regulating AM development and function. Initiation and coordination of the lung inflammatory response. Lung insults, including infections, initiate in the best case a self-limiting host response that neutralizes or eliminates the source of damage, e.g., the toxin or pathogen, and promotes full regeneration of the damaged tissue to restore quiescence and full lung functionality. The immune response was initially viewed as engaged mainly in pathogen or toxin recognition and removal, and while more recently its involvement in reestablishing tissue homeostasis is increasingly appreciated. Although the host response is tightly regulated to avoid excessive tissue damage and immunopathology, these circuits do not always succeed in calibrating the response appropriately. In this section, we will discuss how immune, epithelial, and stromal cell types individually and through crosstalk contribute to both initiating and coordinating the inflammatory response. Epithelial inflammatory responses. The epithelium is the primary target of many respiratory viruses, allergens, and toxins. While commonly considered just a structural component of the lung, the epithelium also plays a key role in the early response to these insults via the rapid production of cytokines, including alarmins and chemokines, to initiate inflammatory and immune responses. Early in inflammation, epithelial cells represent an important source of IL-33. A member of the IL-1 superfamily, IL-33 is a multifunctional cytokine involved in a variety of different biological processes, including development and regulation of the immune system, as well as tissue homeostasis and remodeling. At steady state, IL-33 is constitutively expressed at high levels and is localized in the nuclei of various stromal cell types, including endothelial, epithelial, and fibroblast cells. IL-33 does not contain a classical signal sequence and therefore is not secreted like a conventional cytokine. Instead, it functions as an endogenous danger signal or alarmin released after cell injury to alert the immune system of tissue damage during trauma or infection. By stimulating a broad range of cells, including T helper 2, Th2 cells, eosinophils, basophils, mast cells, and ILC2s to produce IL-4, IL-5, IL-13, and amphiregulin, IL-33 regulates a cytokine network that both controls pathogen removal and supports tissue repair. At baseline, epithelial and ECs are the predominant sources of IL-33 in the lung, while expressing generally low levels of ST2, the IL-33 receptor. However, under prolonged inflammatory or stress conditions, the expression of both IL-33 and ST2 on non-hematopoietic cells increases, resulting in an amplification of the IL-33 ST2 driven signal in both an autocrine and paracrine matter. Autocrine and paracrine matter. Potato. Type 1 and type 3 interferons are primarily induced by intracellular sensing of viral infections and, in turn, stimulate expression of a wide set of interferon-stimulated genes, many of which encode antiviral activity. Since the receptor for type 3 interferons is predominantly expressed by epithelia, these interferons act almost exclusively in a closed circuit limited to epithelia. In contrast, type 1 interferons are also potent immunostimulators with broad activity on immune cells, promoting innate and adaptive immune responses. In this way, type 1 interferons have a strong potential for tissue damage, and therefore, mechanisms for tight regulation of this pathway are in place. Insufficient regulation leads to excessive innate immune-mediated tissue damage or exhaustion of adaptive immunity. Endothelia in inflammation. Compared with epithelia and leukocytes, less is known about the role of endothelium during inflammation, even though it is an equally important component of the alveolus, whose barrier function relies on maintenance of both the epithelial and endothelial layers. While most viruses do not directly infect the endothelium, the robust inflammatory response induced by these pathogens causes significant injury to the pulmonary vasculature. This vascular disruption can cause loss of barrier function and pulmonary edema, accumulating in acute respiratory distress syndrome, a complication of viral pneumonia that can result in multi-organ failure and was a major cause of lung damage and mortality in the COVID-19 pandemic. Endothelial responses to inflammatory stimuli can be both rapid and sustained. Rapid responses, known as type 1 activation, do not require de novo gene expression, but rely on a release of pre-synthesized molecules, including the leukocyte adhesion protein P-selectin, the neutrophil chemoattractant CXCL8, and the prothrombotic molecule VWF in response to ligands that typically bind G protein-coupled receptors. In contrast, type 2 responses represent the classical response to pro-inflammatory cytokines, namely to IL-1 beta and TNF-alpha, resulting in activation of transcription factor NF-kappa beta and AP1, and in de novo protein synthesis of molecules such as vascular cell adhesion molecule 1, VCAM1, intracellular adhesion molecule 1, ICAM1, and CCL2 that favor a transition from the initial pro-neutrophil to a pro-mononuclear type of cell activation and extravasation. Both type 1 and type 2 activation cause increased blood flow, vascular leakage of plasma proteins, and leukocyte recruitment at sites of inflammation. While type 1 activation is short-lived due to receptor desensitization, type 2 activation can persist and amplify as long as inflammatory cytokines are present. Over time, immune cell-derived cytokines may also contribute to and exacerbate EC activation. TNF-alpha and IL-1 beta, in combination with interferon gamma, can trigger EC death with exposure of the thrombotic basement membrane and initiation of a clotting cascade, which can amplify into systemic coagulation and thrombosis, a phenomenon termed thrombotinflammation, which is frequently observed in severe COVID-19. Type 1 interferon responses also have been associated with endothelial dysfunction by reducing nitric oxide availability and by skewing the profile of endothelium-secreted molecules towards a prothrombotic state, while inhibiting normal angiogenic functions that may be relevant for respiratory processes. In addition to being regulated by cytokines, ECs can also produce them. In a murine model of H1N1 2009 pandemic influenza virus infection, activation of endothelial S1P signaling by a synthetic agonist suppressed early innate immune responses and improved animal survival. The orphan hormone receptor / transcription factor COOP-TF2 was also shown to play a role during influenza infection in regulating EC proliferation and migration through direct transcriptional activation of the cell cycle regulator cyclin D1 and potentiation of VGFA/VGGFR2 signaling through NRP1 neuropilin-1 activation. Activation of NF-kappa beta by pro-inflammatory cytokines such as IL-1 beta and TNF-alpha inhibited COOP-TF2 activity and subsequent repair. Thus, although essential for orchestrating protective responses against viral infection, chemokine and cytokine production, even in the context of ECs, needs to be strictly regulated to prevent morbidity and mortality. Finally, Activity of the environmental sensor aryl hydrocarbon receptor in ECs during respiratory virus infection induces a transcriptional response that mitigates epithelial damage and promotes barrier integrity. Innate immune cells in pulmonary inflammation. Apart from having a role in maintaining lung homeostasis, AMs play important roles in recruitment and activation of a variety of cell types, including monocytes, neutrophils, and dendritic cells, via the production of a plethora of pro-inflammatory cytokines and chemokines. IMs also contribute to cytokine responses by producing immunoregulatory cytokines such as IL-10 and the IL-1 receptor antagonist. Indeed, mice deficient in IMs exhibit enhanced cytokine and chemokine production in the context of low-dose PR8 H1N1 infection. This picture is, however, complicated by the observation that the AM pool often undergoes rapid depletion upon both viral and bacterial infection, or in other inflammatory contexts, such as bleomycin administration. This phenomenon, termed the macrophage disappearance reaction, may occur in a highly coordinated and rapid manner in response to insults. The exact cause and potentially purpose of this phenomenon, which has also been observed in other tissues such as the liver, remain elusive and may be a result of inflammation or a byproduct of high pathogen burden, or maybe due to damage to the niche, which is critical in instructing and maintaining AM identity. Indeed, the macrophage disappearance reaction is common in infections by viruses, which target, thrive in, and damage the airway and alveolar epithelial cell compartments. It has also been postulated that the tolerizing program of AMs/macrophages, maintained by continuous phagocytosis of apoptotic cells, may be incompatible with the pro-inflammatory signaling needed to clear incoming pathogens effectively. Thus, this response may represent an example of cell-intrinsic altruistic behavior by enhancing the speed and amplitude of recruitment of monocytes and neutrophils that may be more effective near the infection. Inflammatory monocytes represent an important source of inflammatory cytokines and chemokines, as well as a source of reactive oxygen intermediates and reactive nitrogen intermediates in conditions of inflammation. Recruitment of monocytes into the inflamed lung is typically primarily driven by their expression of CCR2, with lung epithelial cells and monocytes representing important sources of both CCL2 and CCL7 early and later in infection, respectively. The presence of inflammatory monocytes during infection can be advantageous or harmful, depending on the context, and involves a fine balance. Trail-expressing monocytes and macrophages promote death in DR5-expressing epithelial cells during influenza infection through trail DR5 interactions, exacerbating damage and pathology. Even in mild influenza, monocyte-induced tissue damage can contribute to barrier disruption that facilitates bacterial superinfection, as CCR2 knockout mice are more resistant than wild-type to post-influenza bacterial superinfection, but not to single bacterial infection. Another crucial role of infiltrating monocytes is to contribute to the recovery of the AM niche by differentiating into bone marrow monocyte-derived AMs, BMAMs. The contribution of BMAMs to the AM niche after lung insult is once again context dependent. In some cases, resident AMs may suffice to repopulate the vacant niche. In others, bone marrow AMs may contribute to this repopulation and engraft in the lung, self-renewing long-term. The decision between local proliferation and repopulation with monocyte-derived macrophages is likely insult-dependent and influenced by the cytokine milieu driving the inflammatory response. This is highlighted by the difference in capacity for monocytes to give rise to long-lived monocyte-derived macrophages in the pleural cavity across different mouse strains. Neutrophils are another key component of the inflammatory response during disease. They are often the first cell to be recruited to the infected lung and can be activated via several signals, including cytokines, chemokines, and pathogen and damage-associated ligands. Furthermore, the process of neutrophil extravasation and diapedesis can potentially activate neutrophils. In response to such cues, neutrophils contribute to pathogen entrapment and clearance via phagocytosis and the production of neutrophil extracellular traps, nets, and the decision between these strategies is determined by pathogen size. Nets can also propagate the immune response, as they can be internalized by dendritic cells and macrophages, activating cGAS, cyclic GMP-AMP synthase, and therefore type 1 interferon production. Neutrophils can also promote antimicrobial killing through the production of myeloperoxidase and neutrophil elastase, which catalyzes ROS, reactive oxygen species, production, while also catalyzing proteolysis. Matrix metalloproteinase MMP expression can also facilitate neutrophil migration through the ECM. Neutrophils produce a variety of chemokines that can recruit more neutrophils, monocytes, dendritic cells, and various T cell subsets, in addition to producing a plethora of pro- and anti-inflammatory cytokines that can influence cellular activation states. For example, during Nipostrongylus brasiliensis infection, neutrophils can adopt a type 2 transcriptional profile and secrete IL-13, which signals macrophages to adopt an anti-parasitic functional profile. However, there is a fine balance involved in these processes, as excessive degranulation, netosis, and pro-inflammatory cytokine production can equally contribute to disease exacerbation. While neutrophils are equipped to contribute to the resolution of microbial infection, it is less clear whether they are beneficial in respiratory viral infections, as recruitment is virus strain dependent and neutrophil depletion can have some variable outcomes in terms of disease severity and in different models. Indeed, severe diseases caused by SARS-CoV-2 is characterized by exaggerated neutrophil response and the emergence of immature neutrophil population in the bloodstream and lungs. And NETs can induce thromboinflammation by acting as a scaffold for activated platelets. Neutrophilic inflammation appears to also predispose to RSV infection in human experimental challenge studies. Interactions between immune, epithelial, and stromal cells during inflammation. In addition to orchestrating local immune cell recruitment through the release of soluble mediators, epithelial cells can directly influence barrier immunity through cell-to-cell contact. AMs are highly migratory, but during LPS-induced lung inflammation, a population of AMs remain sessile and communicates with the alveolar epithelium in the form of calcium waves via connexin 43 containing gap junctions, which causes CAMK alpha-dependent phosphorylation of epithelial AKT, limiting inflammation. AMs may also secrete SOX1 and 3 in exosomes and microparticles, which inhibit STAT activation, and therefore diverse TLR signaling pathways in alveolar epithelia. In response to Legionella infection, AMs produce IL-1, which induces epithelial GM-CSF production. Such production polarizes infiltrating monocytes towards a more glycolytic metabolic profile, which results in increased monocyte pro-inflammatory cytokine production. Epithelial cells also regulate the inflammatory potential of AMs at steady state and in disease through expression of CD200, which, through binding CD200R, limits pro-inflammatory signaling in AMs by inhibiting signaling downstream of ERK, extracellular signal related kinase, Jun N-terminal kinase, and MAP, mitogen activated protein kinase, and therefore inhibits influenza-induced inflammation. Furthermore, epithelium-derived surfactant protein A inhibits the interaction of interferon gamma with its receptor while promoting IL-4 dependent proliferation. The metabolic state of the epithelium may also feed back onto the AMs. In acute lung injury, hypoxia-induced factor 1 alpha, HIF-1 alpha dependent enhanced glycolysis and AK2 cells results in the production of lactate, which polarizes AMs to an anti-inflammatory phenotype that is characterized by higher IL-10 and reduced IL-1 beta and IL-6 production. Finally, in mice, following acid inhalation-induced acute lung injury, the epithelium releases microvessels containing various microRNAs, including MIR-17 and MIR-221, promoting beta-1 integrin recycling and presentation on macrophages, which enhance macrophage recruitment to sites of inflammation. ECs may also impact the AM inflammatory response. During influenza-induced lung injury, ECs secrete the matrix protein SparkCL1, which exacerbates AM pro-inflammatory cytokine production and enhances immunopathology. Returning to full functionality, the regenerative epithelial response and how it is modulated by innate immunity. During the later stages of an immune response to lung insult, a key priority is repairing any damage resulting directly from the insult or from the immunopathology that it precipitated. Although quiescence is at homeostasis, both the airway and alveolar regions of the lung respond efficiently to injury by activating stem/progenitor populations and promoting proliferation of surviving mature lineages. It is now well established that repair of the respiratory epithelium relies on both physical and soluble factor-mediated interactions of epithelial progenitor cells with other cell types, including fibroblasts, endothelial cells, smooth muscle cells, and immune cells. However, by the same token, the presence of aberrantly activated immune or stromal cells may antagonize the repair process, contributing to dysplastic repair, and even chronic damage. In this section, we highlight the important pathways involved in epithelial repair and how these may be modulated by innate immune signaling. Airway and alveolar epithelial repair. Reusing developmental pathways. Following injury, damaged lung epithelial cells are shed from the lining surfaces, leaving a denuded basement membrane. To restore disrupted lung barrier functions, a regenerative process starts immediately after the lesion has occurred. The integrity of underlying tissues and ECM is primary importance in directing repair of epithelial injury, and these scaffolds provide the appropriate niche for achieving correct cellular expansion and differentiation. Emerging evidence shows that proper epithelial repair involves the recapitulation of key developmental pathways involved in organogenesis, organogenesis on a local and controlled scale. Depending on the type and severity of injury, various cell types are recruited to participate in the repair process by re-entering the cell cycle and differentiating into multiple lineages. In addition to airway, basal, and alveolar AT2 cells, whose major role in repair of the respiratory epithelium was already recognized decades ago, other cells present in distinct niches throughout the lung can be activated to self-renew and differentiate, mediating graded and region-specific responses to injury. The airway epithelium is frequently infected and undergoes desquamation and loss upon injury. In response, resident cytokeratin 5 positive, CRT5 positive basal cells proliferate and migrate to cover damaged areas, forming a transient stratified structure. Subsequent differentiation restores lost secretory, goblet, and multiciliated cells and the pseudostratified architecture of the airway epithelium in a process regulated by Notch and MYB signaling. Within the alveolar epithelium, lineage tracing studies showed that, following lung injury, previous quiescent AT2 cells start proliferating for self-renewal and can differentiate into AT1 cells. In the mesenchymal compartments, infection and injury result in the activation and proliferation of various fibroblast types, including myofibroblasts, lipofibroblasts, matrix fibroblasts, and canonical fibroblasts. W and T signaling is a key mediator of cellular crosstalk in the lung. After viral infection, a subset of W and T responsive AT2 cells, named annexin-2 positive AT2 or alveolar epithelial progenitors, AEPs, is responsible for the majority of AT2 cell proliferation. APEs are major contributors to functional alveolar regeneration and respond robustly to both fibroblast growth factor, FGF, and WNT signals. FGFR2 activation promotes AT2 cell proliferation, while WNT signaling modulates the AT2 to AT1 transition. During efferocytosis, AMs can contribute to Wnt signaling by producing TRAFL factor 2, which in an autocrine manner promotes the production of Wnt ligands. Interestingly, Wnt signaling in macrophages promotes a pro-inflammatory phenotype through a beta-catenin HIF1 alpha axis, suggesting that Wnt ligands produced during epithelial damage may feed back onto AMs to enhance a pro-inflammatory program. Broad Wnt activity in the alveolus could, however, be detrimental and promote fibroblast activation and expansion. In a recent elegant paper, the precise mapping of Wnt receptor expression in the alveolus and the development of frizzled-specific agonists allowed the specific targeting of frizzled receptors on AK2 stem cells, thus decoupling the pro-regenerative and fibrotic effects of Wnt. FGF signaling is a potent mitogenic pathway that drives lung development and injury regeneration. FGFR2 signaling is required for lung branching morphogenesis and for maintenance of AT2 cell fate. After airway injury, hippo signaling increases epithelial Wnt7b expression, which in turn induces FGF10 expression in the mesenchyme. FGF10 subsequently activates FGFR2b signaling on basal cells to drive their expansion, mobilization, and differentiation. A series of studies showed how FGFR2 signaling is critical for the proliferative responses of both distal lung airway progenitors and AK2 cells. Other studies have also demonstrated a direct link between inflammatory stimuli and downregulated FGF10 expression. In an experimental mouse model of saccular lung development, activation of TLR2 or TLR4 inhibited FGF10 expression, leading to abnormal saccular airway morphogenesis. FGF10 expression inhibition was subsequently shown to involve NF-kappa beta dependent recruitment of the inflammatory factor SP3 to the FG10 promoter after exposure to inflammatory stimuli. During lung development, TGF-beta signaling plays distinct roles in the epithelium and mesenchyme, and is required for the epithelial-mesenchymal interactions that drive lung branching morphogenesis and alveologenesis. After furin-mediated proteolytic cleavage, TGF-beta is initially released into a latent form, which is then activated by serine proteases such as plasmin and MMPs, in addition to thrombospondin and integrins such as alpha V beta 6. Thus, TGF-beta signaling is regulated at multiple levels and influenced by the composition and identity of cells in the microenvironment. TGF-beta has potent anti-prolific effects on a wide variety of cells and inhibits proliferation of AT2 cells. It is also a powerful inducer of epithelial mesenchymal transition, a process in which polarized epithelial cells acquire mesenchymal phenotypes with enhanced motility. During this process, TGF-beta represses adherence and tight junction proteins such as E-coherin and CL1, zonula occludens 1, thus disrupting epithelial cell junctions and apical basal polarity. TGF-beta can also inhibit the expression of AT2 markers and enhance differentiation of cultured AT2 to AT1 cells. In the lung, TGF-beta is produced by a wide variety of cell types, including AMs, alveolar epithelial cells, ECs, fibroblasts, and microfibroblasts. When activated, TGF-beta acts as a pleiotropic growth factor that can induce monocyte and fibroblast recruitment, fibroblast proliferation via platelet-derived growth factor (PDGF) expression, and SMAD3-mediated deposition of collagen and ECM by activated fibroblasts. Immune-mediated impact on tissue repair programs. Type 2 immune responses may best exemplify how direct epithelial immune cell crosstalk can be, with epithelial cell damage activating ILC2 cells that produce cytokines inducing protective epithelial responses. In nematode infections, type 2 responses are triggered to induce epithelial shedding and mucus production and to support repair of the physical damage in lung tissue. ILCs are activated by alarmins, IL-25, IL-33, and TSLP, thymic stromal lymphopoietin, which, as discussed above, are set free by epithelial when they are activated, stressed, or dying. In addition to AT2 cells, cell types that can produce alarmins include endothelia, fibroblasts, smooth muscle cells, and AMs. In turn, alarmin-activated ILCs produce cytokines and growth factors such as amphoregulin, IL-5, and IL-13 that act directly on epithelial cells to drive repair and pathogen expulsion. More generally, ILC2 activation is driven and modulated by a variety of factors, suggesting that ICL2s may be sentinels for the tissue microenvironment in a broader sense beyond a mere response to epithelial stress. Amphoregulin is a member of the epidermal growth factor, EGF, family, which is often but not exclusively linked to type 2 immune responses. Amphoregulin production by ILC2s was initially shown in a murine model of viral infection in RAG-deficient animals. Regulatory T cells, Treg, derived amphoregulin was then identified as important for maintaining lung function during influenza virus infection, although other EGF receptor, EGFR, ligands are abundantly produced by both the epithelium and other immune cells in the lung. A subsequent study proposed a mechanism by which Tregs stimulate the alveolar regenerative niche. T cells accumulate at sites of damage and provide pro-survival cues to a population of collagen 14 positive, Col 14 positive mesenchymal cells. These Col 14 positive cells express high levels of EGFR and are uniquely amphoregulin responsive and support AT2 cell proliferation through FGF7/10 production. Lack of Treg cell-derived amphoregulin causes dysregulated activation and increased apoptosis of Col 14 positive mesenchymal cells and led to a delayed repair response, dysplastic alveolar regeneration, and reduced lung function post-infection. Other immune cells, including Th2 cells in Trichuris marius infection and AMs in Nipah infections or naphthalene exposure, also produce amphoregulin, suggesting its importance in promoting epithelial repair also outside of the context of a classical type 2 immune response. Interesting, after naphthalene exposure, amphoregulin production by AMs was driven by ATP and other cellular components released from dying epithelial cells in a pannexin-1 dependent manner, suggesting that specific mechanisms may relay epithelial damages to AMs to reprogram them to a pro-resolving phenotype. Cytokines can also influence the cellular fate adopted by epithelial progenitor cells. In organoid cultures, various EGF family members support the expansion of basal cells at the expense of both ciliated and goblet cell differentiation, while ciliated cell differentiation is inhibited by Th2 cytokines. IL-3 and the closely related cytokine IL-4, acting through a shared receptor, directly affect the airway epithelium structure by promoting goblet cell generation via the inhibition of FOXJ1 transcription through STAT6 binding to a target site in the FOXJ1 promoter. IL-13 also acts directly on epithelial cells to induce airway hyperresponsiveness and mucus production, two features of asthma, even in the absence of eosinophilic inflammation, subepithelial fibrosis, or emphysema. By contrast, IL-6/STAT3 signaling promotes multiciliogenesis through inhibition of Notch signaling and activation of ciliogenesis-related genes. Interferons, discussed above as potential antiviral and immunomodulatory cytokines, can also impair and delay lung tissue repair through their anti-proliferative and anti-differentiation effects. In viral infections, late or excessive interferon can therefore aggravate disease and facilitate bacterial superinfections, highlighting the importance of timing and duration on the interferon response in infection. Insufficient early interferons may fail to control viral replication, leading to higher virus levels with strong interferon-inducing capacity and consequently abundant late interferons. This timing may explain the apparent contradiction between reports of both low and excessive interferon signatures in severe COVID. In addition to infected epithelial cells, resident dendritic cells and alveolar macrophages are a prominent source of interferons during infection, indicating how innate immune cells can negatively impact epithelial repair via interferons. Innate immune cells can also promote tissue repair. In the context of viral pneumonia, AMs are a source of PLET1, placenta-expressing transcript 1, which ameliorates damage and contributes to restoration of alveolar and barrier integrity by promoting proliferation of AT2 cells, as well as enhancing expression of tight junction associated molecules with the alveolar epithelium. Co-culture experiments of AT2 cells and AMs have also shown that AMs promote the proliferation and survival of AT2 cells in a minimal system. The lung environment during repair can also decrease the pro-inflammatory potential of macrophages through the release of diverse lipid mediators, including lipoxin, resolvins, and protectants at later stages during infection. Other macrophage populations may also play important roles in supporting epithelial repair. Short-lived cMAF/MAFB dependent Ly6G positive population of monocyte-derived macrophages, recently described in the context of influenza infection, promotes AT2 mediated alveolar regeneration. Furthermore, in a model of poliodosinal induced tracheal injury, a population of intraepithelial macrophages was identified that is lost after injury and replaced by CCR2 positive monocytes. Interestingly, in global absence of CCR2, repair of the tracheal epithelium is impaired, suggesting a role for monocyte-derived macrophages in the regenerative process of the airway epithelium as well. Researchers recently showed in the context of influenza infection that IMs are key players in the clearance of infected AT2 cells during the resolution phase of influenza infection. Overall, recent work has started to reveal that increasing complexity of crosstalk between innate immune response and lung epithelial cells, ECs, and mesenchymal cells during tissue damage and repair. Better insight into these interactions will further our understanding of what determines the severity of lung insult and open novel pathways for intervention. Long-term impact of acute infection, from memory to scar. Even when the response to a lung insult is self-limiting and return to quiescence is achieved, the new steady state may not be the same as the original one, with positive or detrimental consequences. Here we discuss lasting changes to both the immune and stromal compartments that extend beyond the end of the original insult and the acute response to it. Long-term imprints in immune cells. Perhaps the most striking example among immune cells is changes in the AM population. Lung infection or injury can result in the emergence of bone marrow AMs, which engraft in the lung long-term and exhibit altered functionality in terms of cytokine responses to ex vivo and in vivo re-challenge, metabolic status, and proliferation. Interestingly, some of these features, such as high production of IL-6, mirror features of their monocyte precursors, suggesting that they may be retained during the differentiation process in a phenomenon previously termed monocyte legacy. The presence of bone marrow AMs, in keeping with their altered functionality, can influence the outcome of future infections. While beneficial in the context of Streptococcus pneumoniae infection, BMAMs are detrimental in the context of PR8 infection. BMAMs also exhibit an enhanced proliferative capacity that may thus outcompete their resident AM counterparts and overcrowd the alveolar niche. While recruited BM-AMs progressively acquire features of resident AMs over time, this process is slow, and the mechanisms and tissue signaling that drive this adaptation remain to be elucidated. The AM compartment may also be altered functionally through long-term imprints in the resident AM compartment, as observed in the context of adenovirus infection and low-dose LPS exposure. Importantly, the recruitment of BM-AMs and the training of resident AMs has never been observed at the same time. Care must be taken to exclude that functional changes at the population level are not just the consequence of BM-AMs that have joined the AM pool and contribute to new functionality in bulk analysis. Appropriate separation of the two populations using genetic lineage tracing approaches, bone marrow chimeric mouse models, and in some cases, in vivo labeling and surface marker expression is paramount in investigating such questions. Finally, long-term changes that improve the subsequent infections have also been linked to tissue-resident memory T cells, TRM-mediated epithelial imprints. Here, pneumococcal pneumonia generates pneumococcus-specific CD4 TRMs that fine-tune lung epithelial cells to increase CXCL5 expression and enhance neutrophil recruitment during lethal heterotypic pneumococcal infection challenge. Long-term imprints in epithelial cells. Studies in the last decade have started to unveil the importance of immune signaling during repair and regeneration and its role in establishing adaptive memories in epithelial cells exposed to transient inflammation or to injury. As already well described in epidermal epithelia, inflammatory triggers, both acute and chronic, can rewire transcriptional programs, reprogram cell function, and potentially cause permanent and negative changes to tissue functionality. In some cases, the initial trigger can induce changes in a cell's functional state that are maintained after stimulus termination. This has been described for all allergic inflammation in the respiratory epithelium in a comparison of seq-RNA sequencing data obtained from nasal scrapings of healthy control and chronic rhinosinusitis samples. Significant differences were found between non-polyp and polyp basal cells, suggesting that epithelial stem cells may harbor an imprint from allergic memory. But it will be important to assess such differences in epithelia that are less changed in their whole biology than polyps. Technologies allowing lineage tagging of cells that were infected with virus have been developed to test in animal models how surviving previously infected cells are maintained after resolution of the viral infection, and how they differ from their non-infected counterparts. These studies suggest altered transcriptomic states in these cells, but how long they are maintained is unclear. In addition, soluble factors such as interferons may be sufficient to trigger epigenetic changes leading to memory in an epithelial cell, without the need of direct infection of this cell. A remarkable example of an immune modulator capable of influencing epithelial behavior is IL-1 beta. IL-1 beta was first shown to affect growth of alveolar type 2 cells in both in vitro organoid cultures and in vivo after viral injury. More recently, in a model of bleomycin damage, IL-1 beta activated alveolar type 2 cells to generate a population of transient proliferating progenitors that could then differentiate into type 1 alveolar cells. Persistent IL-1 beta signaling, however, blocked these cells from fully differentiating. IL-1 beta also suppresses Notch ligand expression in ciliated cells, causing downregulation of Notch activity in adjacent secretory cells and promoting their conversion to an alveolar fate. These airway-derived alveolar type 2 cells contribute to repair but remain functionally and epigenetically distinct from the resident type 2 alveolar cells. A similar dysplastic expansion of airway-derived cells is induced by excessive IL-22, which stimulates the proliferation of an intralobular subset of secretory cells into both the injured airways and alveoli. Following severe respiratory infections, the substantial loss of alveolar type 1 cells could lead to impaired gas exchange. The resultant hypoxia triggers activation and recruitment to the distal lungs of rare SOX2 positive, P63 positive airway epithelial progenitors, characterized by KRT5 expression. KRT5 plus positive cells are likely to act in concert with APEs. KRT5 cells respond rapidly to prevent immediate loss of epithelial barrier, while AEPs simultaneously regenerate functional alveoli. The expansion of KRT5 expressing cells into the alveolar region in areas of severe damage to form clusters, pods, was promoted by hypoxia-induced Notch signaling and caused tissue keratinization and consequent long-term respiratory dysfunctions. Local activation of the Wnt/beta-catenin pathway counteracted this dysplastic repair by promoting differentiation of KRT5 cells into proper type 2 alveolar cells. Similar KRT5 pods have also been found in acute human lung injury. Overall, dysregulation of Notch and Wnt signaling, imbalance of TGF-beta signaling, or the presence of a pathological cytokine milieu may lead to aberrant epithelial responses to severe injury and may cause bronchiolization of the alveoli. This process can be described as an emergency repair system to prevent loss of tissue integrity based on the mobilization of cells mainly derived from the airways. In most cases, however, at the end of the process, these dysplastic cells maintain a memory of their original niche and do not fully express the canonical alveolar epithelial markers or reconstitute a functional alveolus. Chronic inflammation. The purpose of an immune response is classically thought to be to efficiently identify, respond to, and eliminate incoming insults. Equally important, however, are the resolution of the immune response and the coordinated restoration of tissue integrity and function. Resolution involves precise integration of signals with the structural and functional cells of the tissue. However, there are cases in which the return to baseline goes awry, whether due to chronic insults, persistent pathogen, host comorbidities, genetic predispositions, or synergies between these factors. In these cases, the feedback between innate immunity and structural cells that previously drove a cascade toward resolution can drive a cycle of persistent and excessive inflammation, culminating in chronic lung disease. There are many chronic lung diseases, each with diverse causes and mechanisms, including asthma, pulmonary fibrosis, COPD, and chronic infections such as Mycobacterium tuberculosis infection. In this section, we highlight the role of cellular crosstalk in chronic inflammatory lung disease with a focus on pulmonary fibrosis. However, the principles described here feature in many, if not all, chronic lung diseases. Pulmonary fibrosis is a condition associated with advanced stages of interstitial lung disease, which may result from individual or synergies between genetic predispositions, age, and single or recurrent environmental insults, such as infections. It may also arise with unknown etiology, in which case it is termed idiopathic pulmonary fibrosis. Pulmonary fibrosis involves persistent alveolar injury with aberrant repair and an excessive fibrotic response. Over time, this results in fibrosis, which is characterized by excessive extracellular matrix deposition with foci of microfibroblasts, fibroblasts, and collagen accumulating and impairing proper physiological lung function, including ventilation at the large airways and gas exchange at the alveoli. The only existing treatment option for unresolved IPF is lung transplantation. Cytokines, growth factors, and matrix remodeling factors constitute a primary mechanism of both direct and indirect intracellular communication within a tissue, and several of these have been implicated in pulmonary fibrosis. A key cytokine contributing to the fibrotic process is TGF-beta. It acts on multiple cellular processes, including epithelial cell proliferation, alveolar epithelial cell differentiation, fibroblast activation, and extracellular matrix organization, all closely associated with pathogenic tissue remodeling in pulmonary fibrosis and emphysema. Both alveolar macrophages and alveolar type 2 cells are major sources of TGF-beta at fibrotic foci. Macrophages are also a source of osteoponin, which can activate TGF-beta. Indeed, osteoponin-producing macrophages have been identified in patients with IPF. TGF-beta can stimulate expression of pro-inflammatory and fibrogenic cytokines such as TNF-alpha, PDGF, IL-1-beta, and IL-13, amplifying and perpetuating the fibrotic response. Macrophages can also produce growth factors such as PDGF as well as IGF-1, insulin-like growth factor 1, and profibrotic cytokines such as CCL18 that cause fibroblast activation. Indeed, supernatants from alveolar macrophages derived from IPF patients caused increased collagen production by lung fibroblasts, particularly CCL18 mediated. In addition, macrophages can produce extracellular matrix remodeling components such as fibronectin, matrix metalloproteinases, MMPs, and MMP inhibitors such as TIMP1 and TIMP2, which can be advantageous or deleterious depending on the MMP and the tissue context. Moreover, epithelial cells may contribute to the recruitment and activation of monocytes and macrophages, which exacerbate fibrosis pathogenesis. In the Hermansky-Puldock syndrome, HPS, model of fibrosis, alveolar epithelial cells produce elevated CCL2, which results in recruitment of monocytes that give rise to macrophages producing high amounts of TGF-beta. This fuels a feedback loop that culminates in alveolar epithelial cell apoptosis and fibrotic remodeling. In the context of bleomycin-induced fibrosis, apoptotic non-hemopoietic stem cells release CXCL12, which results in the recruitment of atypical monocytes known as SAT-M, segregated nucleus-containing atypical monocytes, promoting disease progression. In the same way that epithelial development pathways may be co-opted for wound repair, such pathways also go awry in the context of fibrosis. Bronchialization of the distal airways, characterized by the presence of KRT5 cells, has also been observed in fibrosis, in addition to culmination of transitional alveolar epithelial cells expressing both alveolar type 1 and 2 markers. This may in part be driven by the increased IL-1 beta observed in IPF patient bronchoalveolar lavage, potentially originating from monocytes or macrophages. This phenomenon is also potentiated by the increased Wnt signaling observed in fibrotic lungs, which promotes alveolar type 2 self-renewal and impedes indifferentiation. Indeed, Wnt inhibition ameliorated fibrosis in a mouse model. In keeping with the enhanced epithelial self-renewal observed in fibrotic lesions, increased nuclear gap has also been observed in epithelial cells in the context of IPF, together with profound and early Notch reactivation. Thus, proper cell differentiation, essential for reconstruction of the physiological and functional lung niche, is impeded, which results in the accumulation of aberrant epithelial cell populations, further promoting the fibrotic process. Finally, sustained AREG expression in type 2 alveolar cells can drive lung fibrosis through EGFR activation within fibroblasts. The presence of aberrant or senescent alveolar epithelial cell populations not only enhances a fibrotic cellular environment, but may also result in loss of proper immunological niche for resident macrophages. Combined with the increased expression of monocyte chemoattractants, this may contribute to a vicious cycle whereby monocytes are recruited into the fibrotic lung but are unable to find their homeostatic niche. This process can result in either continual monocyte recruitment or differentiation of monocytes into monocyte-derived macrophages, in many cases with fibrotic properties, a phenomenon observed in diverse models of pulmonary fibrosis. There is also heterogeneity within the macrophage compartment in fibrosis. Using RNA-seq, researchers identified a CX3CR1 Siglet F positive transitional macrophage population that exhibits pro-fibrotic properties such as PDGF-AA production, which promotes fibroblast proliferation, and this population localizes to foci of fibrosis in vivo. This observation may reflect the increased epigenetic permissiveness observed in monocyte-derived cells that could result in different phenotypes adopted by monocyte-derived macrophages, even within a single tissue, depending on whether the niche at the monocyte's destination is healthy or fibrotic. Concluding remarks. Recent progress in understanding the impact of immune response on lung tissue regeneration highlights that the immune versus non-immune categories become ever more blurred. Immune cells, like macrophages and T cells, have important functions in tissue homeostasis and repair, and non-immune cells, such as epithelia and endothelia, have key roles in sensing pathogens and damage, and in recruiting and activating immune cells. Furthermore, the immune functions of structural cells are strongly organ-specific. There is considerable spatiotemporal pleiotropy at both the cellular and molecular levels, and feedback loops must be tightly controlled to prevent excessive and uncontrolled inflammation and regenerative processes. Thus, future work applying more time-resolved, granular, and interaction-based systems-level analysis will pave the way towards not only a better understanding of cellular crosstalk, but also more precise and effective pharmacological intervention to prevent and treat diseases of controlled inflammation and repair. The end.